The reason why that's so thin is it's just sort of like an anchor to make sure this doesn't shift around inside the chloroplast. Okay, so this is actually what chlorophyll is doing, okay? So inside chlorophyll, there are a series of electrons, and specifically there's really 2 electrons that we're really interested in here. These electrons, what we are trying to do is we are trying to give potential energy to. So the way that this works is that light is going to strike the chlorophyll molecule in its big radar-shaped head, or satellite dish-shaped head. And what's going to happen is that light is going to bounce around inside the chlorophyll molecule. It's going to bounce between chlorophyll molecules. And eventually, it's going to end up, that energy is going to end up exciting 2 electrons by giving them potential energy. Now, these electrons don't come from anywhere. What they come from is the splitting of water, or the hydrolysis of water. So we're basically, actually, it's probably better to just call it splitting of water more than hydrolysis, just to keep our terminology the same. So we're going to splitting of water, and what happens when we split water is we release oxygen as a byproduct. We like this. This is what we breathe. It also gives us some protons. Protons we're going to use a little bit later on in the light reaction. But at this step, what's important is that it coughs up these electrons. These electrons are what we're going to use in order to store potential energy. This potential energy that's ultimately stored in an electron is going to give off some heat. It's going to give off some photons. We're not too worried about the first 2 here. But the potential energy that's ultimately stored in these electrons is eventually going to be used to make ATP and NADPH. All right. So here's the overall idea here, is you have some energy in the form of light coming in, hits a chlorophyll molecule, that energy bounces around the chlorophyll molecule, and eventually that energy is transferred to a pair of electrons that sit in the middle of a very specialized chlorophyll molecule. We'll come back to what that is in just a second. And the idea is that once that light energy gets funneled towards the electrons increase their potential energy. So we're temporarily storing energy inside these electrons. And then we can use that energy stored inside the electrons to make ATP and eventually to make NADPH. Where we put our chlorophyll are something called photosystems. And in the light reaction, there are 2 photosystems. They're called photosystem 2 and photosystem 1. These are basically large protein complexes. They store lots of chlorophyll molecules. They also contain these specialized electrons that we're going to add our potential energy into in order to get them to make ATP and NADPH. Regardless of whether you're talking about the first photosystem or the second photosystem, these proteins break down into sort of 2 larger parts. The first is the reaction center complex. This is where a very specialized pair of chlorophyll molecules hang out. This is also where our electrons that we're trying to drive potential energy to also hang out. So in the reaction center complex, ultimately what we're trying to do is transfer light energy to the reaction center complex so that the electrons sitting in the reaction center complex gain that energy in the form of potential energy. Otherwise put, this is where this pair of electrons that we're trying to excite hangs out. If we relied on just a pair of chlorophyll molecules to pick up a lot of light energy, it'd be a really inefficient system. So what we do is we surround that specialized pair of chlorophyll molecules that sit in the reaction center complex with a ton of other chlorophyll molecules. So we're increasing the surface area from which we want to use to capture light. So the second part of our photosystem is called the light harvesting complex. It surrounds the reaction center complex and it's rich in chlorophyll molecules. We do this, again, to try to increase the amount of light energy that are captured inside this complex. Ultimately, whether you get the light in the light harvesting complex, wherever you get it, wherever that light is captured, it's ultimately funneled to the reaction center complex to excite this pair of electrons. All right, if we back up here, all the way to our picture of this guy right here. Remember, these guys here are called thylakoid discs, right? Stack them up, create this stack of granite. Why do you think this has like folds on the inside of it? Why not just be a hollow tube? What do you think the cell's trying to do here?
Increase surface.
Yeah, it's exactly what it's trying to do. It's trying to increase surface area. Do you guys remember mitochondria? It's got all the extra folds in the inner membrane, increasing surface area. It's the same thing here that this chloroplast is trying to do. And the reason why it does it is that these photosystems, where we're trying to capture light energy, they hang out in the thylakoid membrane. So the more thylakoid membrane you have, the more photosystems you can put in it, the more light energy you can capture. All right, questions so far? All right, so each light reaction contains two different photosystems. Photosystem 1 and photosystem 2. Now the thing that would always screw me up in Bio 1 is the fact that photosystem 2 comes before photosystem 1 in the light reaction. And the only reason why that is, they found photosystem 1 before they found photosystem 2, and then they figured out the order in which it was. So they kind of screwed it up, but so that they don't have to go through literature and correct everything, they just left it as is. So keep in mind, photosystem 2 always comes before photosystem 1. Okay, this is what a photosystem looks like, or at least a cartoon of it. Again, we have our light harvesting complexes, shown in purple here. And in the center, there's the reaction center complex. These electrons that we're ultimately trying to drive energy towards sit in the center of the reaction center complex. This is the thylakoid membrane right here. Again, this is all folded over, trying to increase surface area, trying to maximize how much light the cell can capture. In the light harvesting complex, you have lots and lots of chlorophyll molecules. And again, the idea here is that when light strikes one, that energy is going to be funneled to the next chlorophyll molecule, that's funneled to the next, that's funneled to the next. And ultimately, we're trying to funnel that light energy into the reaction center complex, where we have a specialized pair of chlorophyll molecules where these electrons sit. Okay, so I'm going to back up here just a second because I don't think I did a good job at explaining this. I'm going to just switch this over where these electrons are coming from. All right, how is this set up today? Okay. There we go. Somebody really likes to sit right. I don't know who they are. All right, so what we do is this. Okay, to get those two pair of specialized electrons, we take water. And then what we do is we send light through it. And water ends up getting split by the energy in light. What we get out of this, oxygen, protons, and electrons. So this is what we breathe. It's a byproduct of photosynthesis. this guy here, we're going to use a little bit later on, but just so it's complete on here, this is going to be used for the ETC, also known as the electron transport chain. And then these guys here, these are our specialized electrons. And these are the ones that we're going to try to transfer light energy to. So this is the one that's going to sit in the middle of the reaction center complex.
The electron transfer chain, does that occur as well as with mitochondria of the plant?
Yeah, so there's two actually. There's a shorter modified one inside the chloroplast, and then there's a longer version of it that we talked about in cell respiration that occurs inside of mitochondria. So we actually have two versions of the electron transport. This one?
Would that one go into which one? Or would it go
This
to
one's
both?
predominantly going to be involved in the one in the chloroplast.
Okay.
What's the carrier for the electrons?
NADPH.
As well?
Yeah. So here, there's no carrier for them. Eventually, these will end up on a carrier. This reaction actually takes place inside the photosystem itself. So the electrons are basically like almost added to the reaction center complex immediately. But eventually, these electrons here end up being carried by NADPH.
Okay.
Yep.
What's the primary electron acceptor that's inside the photosystem?
In this case, the primary electron acceptor. So in this case, there isn't one here per se. In the light reaction, the primary electron acceptor would be NADPH at the end. But it's not something we really sort of like look at and say this is what it is. We keep that sort of stuff over to cell respiration. But in this reaction, that's where they're all funneled to.
What do you put there? Reaction complex?
Oh, reaction center complex
Center.
down here?
Yeah.
Yeah.
Awesome. Bless you.
All right, everybody have this down okay? You guys have all seen Finding Nemo, right? Most of you?
Yeah.
Remember when the turtle launches into the Australian stream there? What's that
Yeah.
called?
Oh.
Whatever it's called. Every time I'm on that damn Nemo ride at Epcot, he's like, let's get into the APC or whatever the hell it is. I immediately think ETC because I'm a big giant nerd. Now you will too if you go there. Okay. So that's where these electrons are coming in. So light bounces around. We're trying to transfer light into these specialized electrons. And that light energy ends up exciting those electrons. It stores potential energy in them. Now we always, in diagrams and that kind of stuff, they're always going to show these electrons as an arrow. It's not like they're like levitating or something like that. This arrow is really meant to show you that they're just gaining energy. So it's not like they like lift off the ground and start floating or something like that. Okay, so both photosystems look like this. There's some minor differences between them, but this is sort of the overall scheme of both photosystem 2 and photosystem 1. Okay. So, what we do is we light up these electrons. We give them lots of potential energy. What we want to do is we want to then pass this energy off, or we want to utilize this potential energy to do one of two things. We either want to make ATP with it, or we want to store that potential energy in NADPH. Because again, we're trying to make both of these energetically rich molecules in order to drive the Kelvin cycle. In photosystem number 2, remember this is the first photosystem during the light reaction, these electrons are given to something called a primary electron acceptor. This is a protein that is going to transfer these electrons, in this case, to a modified electron transport chain. So, after photosystem 2, those electrons are going to be used in a modified electron transport chain. What do we make during electron transport chains? What do we do there? What are we making? Starts with an A, ends with a P, there's a T in the middle.
Is it ATP?
It's ATP. Everybody looks dead this morning. Why do you guys all look dead? You guys tired?
Lab report due
When's lab report due?
today.
Rough weekend. It's Halloween weekend. That's self-induced. It's due Thursday?
Today and Thursday.
Oh, today!
Yeah.
And you guys all showed up.
Today.
Usually it's like a ghost town the day it's due.
Well, it's due today and Thursday.
I just
Okay.
don't fall asleep.
Alright, so see everyone's from Thursday's lab, or vice versa. Everyone here's on Thursday.
Yeah, Tuesday lab is here.
Yeah? Yeah, that makes sense.
Yeah, because we have lab. We can't miss lab.
Yeah, that's true actually.
Yeah.
You guys only have, what are we? You guys only have 5 weeks left.
Yeah.
You only have to do
Don't
this for
remind me.
another 5 weeks. And then you get almost, well actually you almost get a month break, which is
Yeah.
pretty good. So just keep it going for another 5 weeks.
And then you have to do it for
And
another
really
five
it's
weeks.
only 4,
And then you have to do
and
it
really when you think about it guys, it's only 4 weeks and 2 days, right?
Yeah.
Because you have Thanksgiving week, right?
Right.
So think about it, you really have 4 full weeks of this left, and then you get a month off.
I know about it. AC.
Basically, wait a month.
Oh yeah, there's lots of stuff coming up. I'm just saying there's like a light at the end of the proverbial semesters.
Proverbial semester. There's
It
no
could
light
be
in the
dark.
end. You're free.
No. Okay, so back to this. Okay, so what we're going to do is we're going to use the light energy stored in these electrons from photosystem 2 in order to make ATP. Now at the end of this, at the end we've used the energy in these electrons to make ATP. The cell is really smart. It doesn't just give the electrons up. What it does is it reloads those electrons onto photosystem number 1. Then what's going to happen is light energy is going to come in. It's going to ultimately be funneled to that pair of electrons in the reaction center complex. Those electrons are going to gain potential energy. And this time, what we do is we pass the electrons off to NADPH. So the product of photosystem 2 is ATP, and the product of photosystem 1 is NADPH. All right, so kind of have a look at what we just saw, all right? Here's photosystem number 2. Here's photosystem number 1. Linking between these here is a modified electron transport chain. So it still has ATP synthase, it still has electron carriers. We're still going to pump protons against the electrochemical gradient. So it still works the same as a normal electron transport chain. Oops. So over here what happens, right? So we get our pair of electrons when light breaks apart water. And remember, we're going to make some oxygen here. We're going to make some protons. These protons are used in this electron transport chain over here. The oxygen's a byproduct. Again, this is what we breathe. These electrons coming off, what we're going to do is we're going to store those in the reaction center complex right here. So light comes in, bounces around. Eventually that light ends up being funneled to this pair of electrons in the reaction center complex, and they gain potential energy. We take these highly excited electrons and we pass them to a protein that we'll just call the primary electron acceptor. And the goal of this primary electron acceptor is to transfer these electrons to a modified electron transport chain. So in this modified electron transport chain, what we're going to do is we're going to make ATP. But these electrons are valuable. We don't want to give them up. Plant cell's smart. So what it does is it takes these electrons and it recycles them. It puts them into the reaction center complex of photosystem number 1. Light comes in, bounces around, eventually makes its way to these electrons in the reaction center complex. Those electrons gain potential energy and they're passed off to another protein called the primary acceptor. A primary electron acceptor. That protein is then going to take these electrons, it's going to transfer them to a side reaction. We don't have to worry about what that is. But the ultimate product of that side reaction is we store those electrons and we store their potential energy in NADPH. So this is kind of like the final resting position or resting place for these electrons. So photosystem 2 makes ATP. Photosystem 1 makes NADPH.
How much? Like how much of each?
It's 180 NADPH per run of electrons, and I think it's 1.25 ATP per pair of electrons. Something like that.
Okay.
We don't have to worry about the numbers for this guy right here, which is kind of nice. Questions about this setup? Yeah.
Why does it like go through a whole different photosystem just to like add the electrons to the NADPH? Can't it just like do it with the electron transport chain? Like.
So I kind of have two answers. So although we draw these as big purple blobs, there are some structural differences between them. So one of the explanations as to why you might need two photosystems is that the enzymes for NADPH reduction, they may only work with the structure of the photosystem number 1. Same thing with the primary electron acceptor and the electron transport for photosystem number 2. The more sort of straightforward answer is that once you've taken these electrons through the electron transport chain, their energy, the potential energy inside them is essentially spent. It's been spent pumping protons against an electrochemical gradient. So to pass them off to NADPH, you're passing off a pair of electrons that aren't excited anymore, that don't have as much potential energy. So if you think about these electrons as like batteries, both photosystems are charging them up. So that's why you need two. Other questions? Okay, so I also have this little diagram here that kind of talks about how energy is moving here. It's got these people in like hard hats that are smashing things around with massive hammers. But the idea here is that when this person down here hits this with kinetic energy, it basically transfers that energy to this electron in the form of potential energy. And then what we do is we drain that potential energy from that electron and we make ATP. So then we need to transfer energy into that electron again. So in this case here, this guy over here, he's smashing it with a hammer. That's taking kinetic energy and transferring it into potential energy. And then what we do is we just store that potential energy here in NADPH. In reality, obviously, there's no people with hammers in this thing. What these hammers are meant to represent is light energy. So it's all about energy transfer. Light energy is getting transferred into electrons as potential energy. We use that potential energy to make something else, or we store it in something else. Then we have to put it back into that system. Okay. This is sort of the geography of this system. So we talked a little bit about where this is. Okay. These are these thylakoid discs. So this is the membrane. And these photosystems, as well as this modified electron transport chain, they're all inside the thylakoid disc membrane. So everybody write that down, because I've asked that like, I don't know, 7 of the last 10 tests I've asked where this stuff is found.
Wait,
So the photosystems and the electron transport chain for photosynthesis are found in the thylakoid membrane. It's important to remember, though, that plants also contain chloroplasts and mitochondria, so they have this modified electron transport chain in the thylakoid, but they also have the regular old electron transport chain that we saw in the last lecture packet inside their mitochondria. So the electron transport chain sets up in 2 locations inside plants. Electron transport chains can be found in both chloroplasts and mitochondria, or thylakoid membranes and in the inner membrane of mitochondria. Excuse me. Questions? Okay. Two other things that I'd like to point out here. This is the stroma of the mitochondria right here. Excuse me. Not of the mitochondria. Of the chloroplast. Remember, the stroma is kind of like the cytoplasm of the chloroplast. Cells are really smart. What they do here is they're creating NADPH and they're creating ATP inside the stroma. So even though all these guys are in the thylakoid membrane, ultimately these energy-rich molecules that they're creating are both found in the stroma. And the reason why they're created inside the stroma is because the next step in photosynthesis, which is the Calvin cycle, also occurs inside the stroma. So now the cell doesn't have to waste time trying to transport ATP or trying to transport NADPH out of the inside of this thing to funnel it to the Calvin cycle. It's already there and it's ready to go. So ATP and NADPH are created inside the stroma. Both of these energy-rich molecules are going to be used to fuel the Calvin cycle, which also takes place inside the stroma. Questions? Okay. So, there's not a lot of numbers to remember for the light reaction. It's not as easily quantifiable as what cell respiration was like. But on average, we make about 1.25 ATP for every pair of electrons. Or this says per photon absorbed. What that means is every time we make a pair of electrons by splitting water in half, or every time a photon is captured by chlorophyll, we create 1.25 ATP. Any questions? Okay, one last little bit here about the photosystems. This reaction center complex, where these electrons hang out, and where these specialized chlorophyll molecules hang out, are specifically areas called photocenters. So the photocenter is a part of the reaction center complex. We give these photocenters different names. Photosystem 2, the photocenter is called P680. And the reason why it's called P680 is that photosystem is excited the most by wavelengths of light that are 680 nanometers in length. In photosystem 1, the photocenter is called P700. And it's just because the chlorophyll molecules that sit there, they're best excited by wavelengths of 700 nanometers. Yep.
Are they like, because I know that they're like obviously they're photosystem, but are they like enzymes or proteins?
Yeah.
Or?
Yeah, so this is a big protein complex that's
Okay.
embedded inside the membrane.
Okay.
Big, big, big.
Thank you.
You're welcome. So if we go back to this diagram, we've seen this before. I just didn't point out these numbers. Here's photosystem number 2, the specialized pair of electron molecules. The specialized pair of chlorophyll molecules, where the electrons hang out. This is called the photocenter. And the name of this photocenter is P680. Over here in photosystem number 1, again, this pair of specialized chlorophyll molecules where this pair of electrons hangs out. This is called the photocenter here. Again, this photocenter is called P700. It absorbs or gets excited by wavelengths of light of 700 nanometers. questions about the light cycle? So what we're trying to do as a recap in the light cycle, capture light energy, transfer that energy, store it in ATP, store it in NADPH. So we're going to use ATP or we're going to use NADPH to fuel the production of simple sugars. All right. So another B cell video. These things are fantastic. This has more information than what we need to know, but I wanted you guys to visualize this process. It talks about resonance. I didn't talk about resonance in the lecture. You don't have to worry about it. I just want you guys to see this process. You guys see the screens okay? Or you want the lights off?
That's okay.
Okay.
The process of photosynthesis produces ATP from ADP and PI by using the energy from light to excite electrons that are passed along an electron transport chain. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. The following complexes are found in the photosynthesis electron transport chain. Photosystem 2, cytochrome B6F, photosystem 1, ferredoxin NADP reductase, and the complex that makes ATP, ATP synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP and the electrons to form NADPH, and ADP and PI, which combine to form ATP. Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem 2 complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem 2, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. 1 photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem 2 are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient, similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the photosystem 1 complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP reductase, also known as FNR. After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP, NADPH, and molecular oxygen are the final vital products of photosynthesis.
Okay. So again, we don't need to know the enzyme names because we didn't talk about those, but to visualize the overall process, that kind of does, I think, a good job at visualizing this overall process. The one thing I wanted to point out, just in case you watch this video at home, and again, we didn't use this terminology in this lecture, so I won't test you on it. This place here is what they're referring to as the lumen. Your textbook calls it the thylakoid space, but it's basically used interchangeably. So if you're watching this video again, that's what they're referring to right here. It's just sort of this darkened gray area in the middle. But again, we don't use that terminology, so it's not going to show up on test number 3 and surprise me. Questions about the light reaction? Okay. Next, we're going to talk about the Calvin cycle, which is also known as the dark cycle, which is also known as the light-independent cycle. On a test, I'm just going to call this the Calvin cycle, just to keep it simple as possible. The goal of the Calvin cycle is to use the energy stored in ATP and the energy stored in NADPH to make the precursors to make glucose. These precursors are called glyceraldehyde 3-phosphate, or they're abbreviated G3P. You need 2 G3Ps or 2 glyceraldehyde 3-phosphates in order to make 1 molecule of glucose. Every time the Calvin cycle turns, you add 1 carbon into the system. So we'll come back and look at this in significantly more detail in just a second. Each molecule of glyceraldehyde 3-phosphate contains 3 carbons. So in order to get 1 molecule of glyceraldehyde 3-phosphate to form, the Calvin cycle has to spin 3 times. All right, so as I mentioned, this occurs in the stroma of the chloroplast. Again, this is kind of like the cytoplasm of this chloroplast. This is an anabolic reaction because we're putting energy into the system to build something complex. And again, that energy is coming in the form of ATP and NADPH. They're created from the light reaction. So we're back to this diagram here. We've looked at the light reaction over on the left-hand side of the screen. Here's the Calvin cycle on the right-hand side of the screen. The majority of the products that we use here, that we make during the light reaction, are going to be funneled into the Calvin cycle. The Calvin cycle, what it's going to do is it's going to grab carbon dioxide from the atmosphere, and through a process called carbon fixation, it's going to take it out of the gaseous phase, put it into the organic phase. The Calvin cycle is going to utilize the energy in ATP and NADPH to output precursors to make precursors to make glucose. In this case, we're talking about glyceraldehyde 3-phosphate, or G3P. All right, so just like we did when we talked about cell respiration, we're going to talk about inputs and outputs of this cycle. The inputs to the Calvin cycle are carbon dioxide. This is a gas, right? This is what we breathe out as a byproduct of our cell respiration. We need to add energy into the system, and the energy comes in the form of ATP and NADPH. And what we're trying to get out of this, ultimately, is this glyceraldehyde 3-phosphate, also known as G3P, because we can use G3P in order to make glucose. All right, so we're going to balance this in just a second, or add numbers to it. Okay, but this one here, I want to look at the logic first before adding values to it. All right, the Calvin cycle, what it does, every time it turns, it adds one molecule of carbon dioxide into the mix. Or it adds one carbon atom into the mix. Now when it grabs a hold of carbon dioxide, that's in the gaseous form. Again, this is what we're breathing out as part of cell respiration. But we can't use, or plants can't use carbon dioxide in the form of a gas. So what they do is they perform something called carbon fixation, where they take carbon in the gaseous phase and they add that carbon to an organic molecule. So carbon fixation, taking gas form carbon dioxide and adding it to an organic molecule to make it organic carbon. This process of carbon fixation is performed by an enzyme named rubisco. I bolded it because I do expect you guys to remember the name of this enzyme. This is the most abundant enzyme on the planet. Because of how many producers there are on the planet, coupled with how many chloroplasts they have inside them, coupled with the fact how often they have to go through the Calvin cycle, the abundance of this really important enzyme is huge. It is what does the vast majority of carbon fixation on the planet. In fact, people, yeah, so carbon fixation, what you're doing here is you're taking carbon dioxide, which is in the gas phase, so the carbon that's attached to it is also considered to be a gas, and you're taking it. This enzyme rubisco grabs onto it, and what it does is it takes that carbon and it adds it on to an organic carbon. So something that's no longer in the gas phase, but something that has like a string of carbons attached to it. So the process of carbon fixation is taking a gas, specifically the carbon, and adding it onto an already organic molecule. So something that the plant cell can use. Okay, so every time the Calvin cycle turns, it can fix one molecule of carbon dioxide. Glyceraldehyde 3-phosphate, however, is a 3-carbon molecule. That means that the Calvin cycle is going to turn 3 times to make one molecule of glyceraldehyde 3-phosphate. In order to get one molecule of glucose, you need 2 molecules of glyceraldehyde 3-phosphate. That means if you're talking about how many times the Calvin cycle has to turn to make one molecule of glucose, the answer is 6. So the Calvin cycle turns 6 times in order to make one molecule of glucose. Okay. If we take the numbers for the Calvin cycle, and we ask the question, what are the numbers in order to make one molecule of glyceraldehyde 3-phosphate? These are the numbers up here. 3 carbon dioxide, 9 ATP, and 6 NADPH. But what we can do is we can manipulate these numbers based on whether the question's saying per turn of the Calvin cycle, per molecule of glyceraldehyde 3-phosphate, or per molecule of glucose. So, what I want to do here is write that out. So we're over here. Okay. And so this is the Calvin cycle. And here, what we're doing is we're talking about the inputs. So what we'll do is we'll create 3 columns here. The first one will be per term. This will be per molecule of glyceraldehyde 3-phosphate that I'm going to abbreviate G3P. And then the last column here is going to be per molecule of glucose. So what we just saw here is the per molecule of glyceraldehyde 3-phosphates. You need 3 carbon dioxides, 9 ATP, and 6 NADPH. So remember, this is still photosynthesis, so we're still dealing with NADPH. All right, we want to talk about per term. Right, so we're going to scale everything. We have to divide everything by 3, right? Because the Calvin cycle turns 3 times to make 1 molecule of G3P. So if we balance this over here, we get 2 carbon dioxides, 3 ATP, and 2 NADPH. So again, I've divided everything by 3 here, because we only get 1 molecule of glyceraldehyde 3-phosphate if this entire reaction happens 3 times. Per molecule of glucose, we know that we need 2 molecules of glyceraldehyde 3-phosphate. So what we're going to do is we're going to double everything in the center column. So what that means is that we need 6 molecules of carbon dioxide, 18 molecules of ATP, and 12 molecules of NADPH. Yes, these are all the inputs. The outputs are kind of at the top, like 1 molecule of glyceraldehyde phosphate or 1 molecule of glucose. Okay. Questions about how we got to these numbers? All right, let me see here. Why don't we write this at the bottom just for completion? 3 terms equals 1 G3P two G3Ps equals 1 glucose. And that way all the numbers here are just basically on one sheet for you. How many ATP on average do we make during cell respiration?
36.
Right, so we make some like 36 to 38 range, right? So check this out. We only need 18 here, right?
Mm-hmm.
But we make 38 as a maximum output in cell respiration. So the cell is really energy efficient in this process. So here, we're not only using energy from ATP, we're also using energy here from NADPH. But in terms of ATP, there's always a net gain of it inside a plant cell. You're putting in 18 to make a molecule of glucose, but if you take that molecule of glucose and break it down, you end up generating 36 to 38 out of it. So we don't create or lose energy here. We're just always having a net gain in ATP because we funnel our energy towards it, or plants do. Okay, before I knock down the doc viewer here, does everybody have this down okay? Just like every other class, what I'll do is I'll take this sheet. I'm going to put these sheets up at the front. That way, if you didn't get it, you can come take a look at it after class. Okay, so here is what the Calvin cycle looks like. Again, I don't expect you guys to memorize any of the enzymes in here by any means. The idea here is that the input up at the top is carbon dioxide. It's fixed by that enzyme called rubisco, meaning it's taken from the gaseous phase and it's put onto a long organic-based molecule. Then you have a series of enzymes that manipulate the structure of this organic molecule. And at the end of this thing, or at the side of it, you end up creating glyceraldehyde 3-phosphate. What we're using in terms of energy is listed along the side. So here's 6 ATP, over here is 3, so that gives us 9. Here's our 6 NADPH over here. So this, right, as is shown on the screen, is per molecule of glyceraldehyde 3-phosphate. The only thing that I want you guys to know is what we had here upon the sheet, and just this enzyme called rubisco that I bolded on the last slide. But in terms of any of the intermediates here, don't worry about the intermediates on this thing, like 1,3-bis-phosphoglycerate or anything like that. Questions?
What will the NADPH go to?
Oh, so what happens is with NADPH, the energy from the electrons is used to funnel this system. Then what that does is it turns into NADP+. That NADP+ is now ready to accept another 2 electrons, so it will work its way back over to the light reaction,
Okay.
and then it'll get charged up and come back over. So there's a lot of recycling in between the 2 processes.
Okay.
It's the same thing with the ATP that's burned. The ADP that's created gets funneled back over to ATP synthase and the light reaction.
Thank you.
You're welcome. Okay, so it's easier to tell everybody that photosynthesis makes glucose because everybody can grasp what glucose is as opposed to something like G3P. So this glyceraldehyde 3-phosphate, you take 2 of them and you turn them into glucose. You smash them together essentially using an enzyme. This happens as a downstream reaction from the Calvin cycle. So the Calvin cycle doesn't directly generate glucose. It indirectly generates it, or it generates the precursors that we use in order to make glucose. Okay, so what I want to do is just back up here really quickly so that we can see the overall equation here. Right, so we looked at this equation at the start. At this lecture, we also went back and did the same thing when we talked about cell respiration. Because this is all redox reactions, right? We're shuffling electrons around. If we look at this overall reaction, okay? So carbon dioxide is reduced. Remember, this is going to gain electrons. The way that this gains electrons is we take carbon dioxide in the process of carbon fixation and we start adding a ton of electrons to it, right? In order to build glyceraldehyde 3-phosphate, which ultimately allows us to build glucose. So the way that carbon dioxide becomes reduced is we use it to build up glucose. And in that process, we have to add a lot of electrons to it. We need a place for these electrons to come from. Remember, light comes in, it breaks up water to give us protons, an oxygen atom, and this specialized pair of electrons. So in this case here, water is giving up its electrons. And what happens when water gives up those electrons, it's ultimately turned into oxygen over here. Now, of course, we could write things like protons and heat and all that kind of stuff, but just to keep it a little more simple, when we force water to give up its electrons, or we oxidize water, that oxidized water becomes oxygen. So hopefully this makes a little more intuitive sense in terms of why these arrows are connecting the molecular species on either side of this reaction. Questions about the Calvin cycle, the numbers involved in the Calvin cycle, this overall reaction, or the process of photosynthesis? All right, the class average for this test, year over year, is anywhere between 76 and 78. I challenge you as a class to get it to 80. I'm not going to make the test any harder or any easier. I just use the same style of questions I use every year. So I want to see you guys go above 80 and impress me. Because you definitely impressed me on test number 2. You guys do that? Yeah, all right, good. Yeah, go team. You're like, nah, we're going to be in the 72 range. That's what we're going for. I think you guys can do it. I do. If there's no other questions, let's end here for today. We are going to start talking about mitosis next class. Mitosis will not be a part of test number 3.
No, 10. Ajá. 9. ¿Qué era 9? I don't have it. No lo tengo.